Hello everyone, today I'm back with the next video which is on the cell cycle. As some of the students or most of the students they struggle in this topic called the cell cycle or the cell division. So it is very easy to understand and there's nothing hard on it. You just need to understand the concept. So to make it easier, imagine this cell which is in the interface like a student getting ready for exams. It grows, gathers nutrients, and carefully copies its DNA notes. And then comes the big examinations day. So we can consider this big examination day or phase as the mitotic phase, where cell finally divides through mitosis and cytokinesis, making two identical. What type of cells are produced during mitosis? Um, can you guess? You can comment down in the comment box. Okay. I'll tell you further in the next slide what type of cell this is produced. This cycle is super important because it's how living things grow, heal cuts and wounds, and keep their bodies functioning properly. Just like a story that repeats with every new chapter, the cell cycle keeps life going generation to generation. Now, let's talk about the cell cycle. So, when we talk about the cell cycle, what happens is it's such a fascinating journey. So it is primarily broken down into two critical phases: interphase and the M phase. Absolutely, for mitosis, we can break it down by using mnemonics to help students remember. For an example, the phases are prophase, metaphase, anaphase, and telophase. So we can the mnemonic we can build it for it is PMAT. So M phase is the phase where the cell division occurs. Interphase is the phase where cell growth and DNA replication occurs. See, so it is quite different, everybody. So for now, we will be focusing on these two phases only. There are S phase also, and other there is quality control phase also. So we'll focus on it later. Now let's focus on M phase and interphase. So M phase, as I've already told you, is the phase where cell division occurs, and by the end, two daughter cells are formed, and the cycle starts again. So interphase. Now interphase is divided into G1 phase, S phase, and G2 phase. So it is the longest phase of cell cycle. Well, cell grows and prepares for division. So see, there's a big difference. Interphase is the phase where cells get prepares for division. So for an example, the before the examination day you get a holiday for preparations, so that phase you can relate it to the interphase. And M phase is the day of the examination you can relate to. Okay, now what is G1 phase? So S phase was normally in S phase, DNA replication occurred. G1 phase is the growth phase. So as you all can see in this slide, for now we won't be focusing on this one. If you want to know more about G1 phase, I'll make a descriptive video including this also. For now, you can just pause this video and read the slide. Now let's move on to mitosis. As we are moving directly to the mitosis. So what do you think is mitosis? As I've already asked you, like in which type of cells does mitosis occurs, isn't it? So it occurs in, I think you have already got your answer. It occurs in somatic cells. So mitosis is the equational division. For an example, you can relate to when you do photocopy, what happens? There's nothing different. You are just making the same copies of a original document, isn't it? So mitosis is also the same thing. Here, it produces. For an example, let us focus on significance. Then you will more understand what is mitosis. So, it is essential for the growth. For an example, cell division this cell division allows a single fertilized egg to grow into a multicellular organism. Next thing is repair and healing. This is very important. It replaces old, worn out, or damaged cells, which is called healing of a wound, isn't it? Now, reproduction. So, in unicellular organisms, this cell division is the way to reproduce. Now, since as I've already told you, to relate it with the photocopy thing, now come with the genetic stability. It ensures daughter cells get the same genetic information from their parent, so which maintains a stability in that organism. Next thing is it maintains surface area to the volume ratio, which prevents cells from becoming too large to functions, isn't it? Sometimes when cells become too large, they cannot function properly. So it maintains that also. So as I've already told you, to make a mnemonic, which is PMAT, so you can relate to that. The first phase is the prophase, so you can go with it like related to the P for prophase or P for preparations or something, which is setting the stage. So you can just call this phase as the setting the stage phase. As the chromosomes, what happens is they condense and become visible. In this stage, it is the most active stage, as the chromosomes, they condense and become visible, and the nuclear envelope, they dissolve, and spindle fibers, as you can see the top, which is like sun type of figure, it is spindle over there, and their spindle fibers are like thread type of structures, which pulls apart the chromosomes. So spindle fibers begin to form in this phase. So what happens is chromosomes, which are normally spread out in the nucleus, begin to condense and they become visible, as I've already told you. So its microscopic views also show that. So this is a crucial step, like gathering all your materials before starting your project. Simultaneously, nuclear envelope, which houses these chromosomes, dissolves. So this allows the chromosomes to move freely and interact with the next set of structures forming the whole cell. So there are these spindle fibers. So these are like thread-like things. So think of them as, or maybe I could relate it to as the kites beginning to form and prepare for their role of directing chromosomes movement in the next phases. Now, what we can do is, let's highlight the key events. As you can see, you can pause the video, and next thing is you just have to draw chromosomes in this particular figure and appearances of spindle fibers, and dissolving of nuclear envelope occurs in this phase. Now, moving on to the next phase. So in this phase, particularly this phase is metaphase. So M for metaphase, M for middle. I want you to remember the middle word, because in this particular figure, as you can see, the chromosomes, they aligned at the cell's equatorial plane, or sometimes they lie in the middle of the cell. So spindle fibers, they get attached to the centromere, which is like the sun-like structure, which is at the both sides, as you can see. So M for metaphase, M for middle. This will help you remember the metaphase. Now let's go to the next phase, as I've giving you the key events only, so that you could understand. Now next in this phase is the anaphase. Now anaphase, what happens? I want you to remember the word apart. So its key event is what happens is sister chromatids. Now when these chromosomes are pulled apart, now this sister chromatid, now it became sister chromatids. They are pulled apart to the opposite poles by spindle fibers. As you can see, they are pulled apart and they are moving from the spindle fibers towards the centromeres. So each chromatid is now considered as an individual chromosome. Now, moving on to the next phase. So this step is can be considered as an end phase, which is the telophase. Now, I want you to remember the word reconstruction. So, reconstruction means what happens is chromosome now they get decondensed into chromatin. Now, in the prophase, it is totally different now. What happened in the prophase? Chromosomes condense and they start appearing. Now, over here, chromosomes they are decondensing and into chromatin. Now, what happens? Nuclear envelope they reform. See, so you can differentiate between prophase and telophase. In prophase, what happens? Nuclear envelope they got dissolved. Now over here, nuclear envelope they reform around the two nuclei. Now here comes the final division process, the grand finale, cytokinesis. The cytoplasm divides as you can see the cytoplasm they're dividing and they pinches inwards, which we call it like you can see it in the figure also. They're pinching inwards. Eventually, they are separated into cells, two daughter cells, which are diploid daughter cells. Don't get confused. Meiosis is haploid cells, and for this, it is diploid cells. Each daughter cells receive the full set of organelles. They see, so they are completely similar to their parent. They're nothing different. So that's the beauty of mitosis, like making photocopies of the papers. You get the same copy, the original document that you have copied before, isn't it? So this was just the key highlights that I've explained of the mitosis. It is nothing different, everyone. When you read by comparing and different phases